Good morning. Thank you for joining us for our webinar. This month, we're pleased to have Dr. Kerry Sublette, and he's going to be clarifying the question that we have about the differences between compound-specific isotope analysis and stable isotope probing. Now, many of you have probably heard Kerry speak before, and you know that he's a very capable speaker, does a great job of really clarifying some of these difficult questions. I'm really looking forward to this webinar. And although Kerry really needs no introduction, for those of you that aren't familiar, he is the Professor of Chemical Engineering and Geosciences and a Sarkis Professor of Environmental Engineering at the University of Tulsa. Kerry has over 25 years of experience in a wide variety of bioengineering and soil remediation problems. He's got over 13 patents and over 117 publications that range from corrosion to brine spill remediation to ecosystem restoration. So Kerry's really an expert in numerous areas. And he chairs the International Petroleum Environmental Conference and serves on the Oklahoma Board of Environmental Quality. So we're really excited to have Kerry speaking for us today. As many of you have attended before know, the webinar today is being recorded, and it'll be available on the resource section of the Microbial Insights website within the next week. We'll be emailing out a link to all the registrants as soon as it's available. And at the end of this morning's webinar, there's going to be a question and answer period. So as you think about questions during the webinar, please go ahead and type them into the GoToWebinar software, and Kerry will go through and we'll answer those for you at the end. If it becomes too much, then we'll send out an email with some of the answers to those questions if we have to go over time. And if you have questions after the webinar, you can also email us any additional questions to info@microbe.com. And you can also send, if you need certificates, to that info@microbe.com address as well, certificates of attendance for your PDH credits. So without belaboring this any further, I'll go ahead and turn it over to Kerry.
Thank you, Dora. As Dora was saying, we're talking about two stable isotope techniques today. One is stable isotope probing, and the other is compound-specific isotope analysis. What I hope to do today is to clarify the differences between these two techniques and to talk about when one technique might be preferred over another, what the advantages are, what the limitations are. In other words, as the title says here, what's the difference and how do I use them? So let's begin by talking about how they are similar. The applications we'll be talking about today, we'll be talking about how we can use these techniques to actually demonstrate that biodegradation is going on in a contaminated aquifer. Both of these methods are based upon isotopic analysis, mostly carbon-13. And both of them use the same notation and units for expressing carbon-13 content, and that's typified by the equation that you see here on the slide that provides us a definition for what we call the delta 13C of a compound. And the delta 13C is just a way of expressing the amount of carbon-13 compared to carbon-12 in that compound, specifically the ratio of carbon-13 to carbon-12. And it's calculated based on that ratio and the sample compared to that of a standard. The standard is a particular carbonate mineral that everybody uses as a standard. So using this equation, we calculate this del 13C value. The units that we use for this value is symbolized by the little symbol that you see. It looks a little bit like a percent symbol at the bottom there, but the name of the unit is per mil. Now, when we use that equation to define the del 13C values, we end up with a scale. And that scale is shown on the bottom of this particular slide. And on this scale then, if you use that equation to calculate the del 13C value, then the standard, the PD belemnite standard, will have a value of 0. So any compound that contains less carbon-13 than our standard will lie to the left on that scale. And as you can see there, those negative values. Anything with more carbon-13 than our standard would lie to the right and have positive values of del 13C. Also shown on this diagram are ranges of values for common commercial products as well as natural products derived from hydrocarbons. And those are common del 13C values for these materials. And we'll return to that a bit later. So how are they different? Let's first of all talk about stable isotope probing. In stable isotope probing, the key word is probing. We are using a probe, or you might think of it as a tracer, which is a compound, particularly a contaminant of interest like benzene shown here, that is composed totally of carbon-13. And this probe, or this tracer, is then placed in an environment where we think there might be organisms present that have the capability of degrading that compound under aquifer conditions. And if that takes place, then carbon-13 from that original contaminant or probe or tracer is going to get incorporated into biomass and incorporated into products of mineralization. So any carbon compound that's being used as a carbon and energy source, some of the carbon goes into making more microorganisms. That's the biomass you see in the diagram here. And some of that carbon actually gets oxidized all the way to CO2 in the mineralization process, and that's what you see on the bottom here, referenced as the dissolved inorganic carbon. So in stable isotope probing, we're looking for that carbon-13 in the contaminant to end up in one or both of those pools. And if it does, that is proof positive that biodegradation was taking place in the sampled environment under the conditions that exist in that environment. Now, compound-specific isotope analysis is quite different. We're not using any probes. There is no artificial source of carbon-13 or non-natural source of carbon-13 that is being added to an environment. We're looking at the naturally occurring carbon-13 in that environment. About 1.1% of all naturally occurring carbon is carbon-13. So any contaminant that you find in a contaminated aquifer, for example, some of that contaminant is going to have a small amount of carbon-13 in it. Now, when that contaminant undergoes biodegradation, it's a well-known fact that bonds that involve carbon-12, which is the most abundant source of carbon, are slightly weaker than bonds that involve carbon-13. So those bonds involving carbon-12 react faster. So the net result is that during biodegradation, carbon-13 is enriched or accumulates in the undegraded contaminant. So what we're looking for here are changes in CSIA and changes in the carbon-13 content of a pool of contaminant in a contaminated environment. So in a nutshell, those are the differences. We're going to go into the applications of both of these. But just to repeat, in stable isotope probing, we're using a probe or a tracer that is artificially enriched in carbon-13. And we're asking the question, where does that go? And what does it mean once we find out the answer to that question? In compound-specific isotope analysis, we are dealing with only naturally occurring pools of carbon-13. And we ask the question, has the carbon-13 content of a compound in a contaminated aquifer, for example, has it changed over time? And if it has, what does that mean, and what does that tell us as far as the biodegradation of that compound is concerned? So let's specifically talk about stable isotope probing to begin with. As we've already said, stable isotope probing uses a probe or a tracer that is a compound that has been specially synthesized to be composed of carbon-13. And we provide that to microorganisms in the aquifer and give them the opportunity to degrade that compound. Now, how that is typically done is to load that compound into what's called a biotrap. Many of you are probably familiar with biotraps. They're used as passive microbial sampling tools for forensic analysis of microbial communities. It's a product that Microbial Insights markets. The key component are what's called bioset beads that you see in the photo here to the right that are made of 25% Nomex and 75% powdered activated carbon. That powdered activated carbon component is very important to the stable isotope probing method, because what we do is to take that C13 label compound and adsorb it to the activated carbon component of the biosept beads, as illustrated in this diagram, and then we load those beads into the biotrap, and then the biotrap is incubated in the contaminated aquifer. Following incubations, typically 30 to 45 days, the biotrap is going to be retrieved, and then the beads are recovered and they are analyzed. Basically, there are three types of analyses that are typically done. First, we look to see how much of the C13 label compound remains on the beads. The compound that is missing has been biodegraded. Looking at the amount of the C13 tracer or probe that is remaining on the beads can tell us something about, if we compare this to other wells, the relative rates at which the compound is being used in different parts of the aquifer. But most importantly, we look to see where that carbon-13 has gone. We look at the dissolved inorganic carbon pool in the beads. This is going to be carbonate, bicarbonate, CO2 equilibrium in the beads, and we recover that material and look at the isotopic signature of that dissolved inorganic carbon. And we also recover biomass from biofilms growing in the beads and look to see if there's carbon-13 in that biomass. Now we have many options for where to look in that biomass. The most common place to look is in the phospholipid fatty acid component. If you're not familiar with phospholipid fatty acids, these are basically the components that make up the microbial cell membrane. You can think of it kind of as the skin of the microorganism. And carbon-13 will get incorporated into these phospholipids, shown in more detail here, as the microorganism grows at the expense of the degradation of that C13 labeled probe or tracer. So we can see, so there's lots of phospholipid fatty acids in a cell, and so it's very easy to recover these and quantify these and determine exactly how much carbon-13 has been incorporated. Now, I'm going to show you some results in just a moment, a case study, but to take you back to our original scale, remember on this scale, PD belemnite, our standard, has a value of 0, the del13C value. What you're going to see is when you're doing stable isotope probing, if carbon-13 has been incorporated into DIC or into biomass from the probe, the del13C values of the biomass or the DIC will be very far to the right on this scale. My point here is there will be no confusing a positive result from stable isotope probing testing, no confusing that with any naturally occurring pools of carbon-13. The results are going to be quite obvious. So let's do a little case study here. We're looking here at stable isotope probing, evaluating monitored natural attenuation, in this case at an operating refinery where there have been historic releases of gasoline, diesel, naphtha. There is a lot of NAPL present at this particular aquifer. And as you'll see, groundwater here is heavily influenced by a nearby river. This study, we give you a citation here if you would like to read more about this study. The first author here is Kate Key, now Kate Clark. Those of you that have called into Microbial Insights have probably talked to Kate. So in this particular study, it was a very large study, and we're only going to show you just a part of that study. But Kate looked at a number of different, I think it was 26 different monitoring wells at the site, using biotrap samplers and also doing stable isotope probing, where the probe was C13 labeled benzene. Here are the results, or some of the results. from the lipid analysis or phospholipid fatty acid analysis at 3 different monitoring wells, monitoring well 10, 1, and 25. And as you go from 25 to 1 to 10, notice that you're getting closer to the river. On the left-hand side here, that is the del-13C values of the phospholipid fatty acids that were extracted from the beets. Above the bar is also the precise value of the del-13C value. And one of the things I want you to see is how large those values are. There's no environment in which you're going to find del-13C values for any biomass that are going to look like that. So there is no confusion as to what's happened here, that carbon-13 from the probe or tracer has been utilized, has been biodegraded by microorganisms in the aquifer that have the capability of degrading benzene, and they have done that under existing aquifer conditions, and that's resulted in significant incorporation of carbon-13 into the biomass. And as you can see here, that incorporation goes up very significantly as you get closer to the river. If you look at the dissolved inorganic carbon that was recovered from those same biotraps, you see some very similar results. Again, very large numbers for the C13 content of the DIC. If you notice in the far left-hand corner there, minus 20 to minus 30 is a common range for the naturally occurring pool of DIC. So again, there is no mistaking what has happened here. That carbon from that C13 labeled benzene not only has been incorporated into biomass, but it is also some of that benzene has been totally mineralized to carbon dioxide. And once again, you see as you get closer to the river, you're seeing much more utilization of that benzene. So in conclusion, and you read Kate's paper, you'll see that 6 out of the 8 wells where the SIP study was done, there was direct evidence of benzene biodegradation. And as you just saw in that abbreviated data set, the highest C13 incorporation into biomass and the DIC was closer to the river. And other data that you will find in the paper shows that there is a periodic influx of oxygen and nutrients from the river, and that's what's driving the biodegradation process near the river. So if we look at the strengths and weaknesses of stable isotope probing, first, its major strength has got to be that it provides conclusive evidence of current in situ biodegradation activity under actual aquifer conditions. Other advantages, you don't need to know anything about the organisms that are involved in the degradation. You don't need to know anything about the pathway prior to doing SIP. We're just determining whether the target compound is capable of being degraded. You do need to know, though, that the contaminant is likely to be used as a carbon and energy source. Only if it's used as a carbon and energy source will carbon from the tracer be incorporated into biomass or oxidized to DIC and then show up in the analysis. Another advantage is that even with NAPL around, we can readily use stable isotope probing. The NAPL does not interfere at all with this testing. It's a relatively inexpensive method for most common contaminants. Many of the types of contaminants that you see on the list here are readily available in C13 forms and can be used in stable isotope probing. Now, as far as limitations are concerned, if you would like to know what organisms are actually doing the biodegradation, standard SIP will not tell you that, but I did want to tell you that we can increase the C13 content in the biotrap and actually recover and analyze C13 labeled DNA. And with that type of application, we can actually identify what organisms are responsible for the degradation process. Another disadvantage is if you are interested in a very large or complex compound, it could be more expensive to do stable isotope probing because you may have to have that compound custom synthesized to be available for the method. And lastly, and this is one that will lead us into CSIA, as I was indicating earlier, you do need to know that the contaminant of concern, your tracer, your probe, your C13 labeled tracer or probe, is going to be used as a carbon and energy source. This technique is not applicable to contaminants that are used as electron acceptors, like chlorinated ethanes under anaerobic conditions. So that leads us into the topic of compound-specific isotope analysis, because we're going to be talking a lot about compounds that are used as terminal electron acceptors like chlorinated ethanes. Before we get started, though, I wanted to point out a EPA guidance document. If you are going to be using CSIA in your site analysis, if you don't have a copy of this document already, I highly recommend that you get this document. It is an excellent resource. So going back to CSIA and why does it work? And we're going to be taking carbon as our example here. As indicated here, carbon bonds with the lighter isotope, the carbon-12, the one that's the most abundant, that's almost 99% of all carbon, are slightly weaker than those involving the heavier isotope carbon-13. Therefore, they react more quickly. So as it biodegrades, the parent compound then is going to become enriched in the heavier isotope, which increases the del-13C value. The daughter product that's formed, though, is initially going to be very depleted in carbon-13. So it's going to have a lower or more negative del-13C value. This is an excellent illustration that Mike Hyman does, so we borrowed it here. This is C13 M&M's chocolate fractionation, and it's a great illustration of the carbon enrichment process. In this illustration, each individual M&M then represents a quantity or a molecule of a contaminant. And if that contaminant is composed of only carbon-12, then it's blue. If it's composed of carbon-13, then it's yellow, or if it has carbon-13 in it, I should say. And so what is shown as you go left to right, that the contaminant here, the M&Ms, are being consumed. But if there is a preference for consumption of carbon-12, since carbon-12 reacts faster, then there's going to be a preference in utilization of the blue M&Ms. So as you go from the left to the right, notice the number of M&Ms is decreasing, but the proportion of the yellow M&Ms is also increasing. So that's analogous to what's happening during biodegradation of a naturally occurring contaminant, that its C13 content in the undegraded pool is going to increase over time as degradation takes place. So let's consider something other than M&Ms now. Let's consider PCE. I'm going to show you some results from a laboratory study that are very illustrative. In this particular case, what you're seeing here is the degradation of PCE, and as you'll see, its daughter products as well, in a culture of Dihalicocoides. And what you can see in this particular case is after a lag period here of about 3 days or so, you're seeing significant rates of degradation of PCE. Now, if we were to look at the same time at the del-13C content of the remaining PCE, you would see during that lag time, it's not changing very much. But as degradation starts to take place, what happens to the del 13C value of the PC? It starts to increase. So we say it is C13 enriched. We say that the del 13C value is less negative. We also might say things like the remaining PCE is getting heavier because the C13 content is increasing. So this effect can provide qualitative proof that biodegradation is taking place. But the question you might have is, well, how much do you have to see that isotopic signature change before you can be sure that you are looking at biodegradation? And there is a rule of thumb. If you go back to the EPA document, you'll see where this comes from. A rule of thumb is that if we see the del 13C value change by 2 per mil, become less negative, then that is taken as a good rule of thumb that if you've seen at least that much change, then that's good evidence of biodegradation. Now, when you're looking at changes in the del 13C value along a flow path, it's the same standard. You see greater than a 2 per mil change along the flow path, that's taken as a good indication of biodegradation. Because as you move along the flow path from the source, the path represents, as you follow that path, increasing time since that contaminant entered the aquifer. So just to illustrate what we're talking about here again is our plot of the del 13C value versus time for PCE. And it started with a value of -28. And as time goes on, as we've seen before, that del 13C value increases. And we've added a threshold value here of -26 that represents a difference of 2 per mil. And as it becomes less negative, heavier, decreases the del 13C value by, increases the del 13C value by 2 per mil. Once we get past that threshold, this is considered to be convincing evidence then that PCE is being biodegraded. And here I've just added now the other, the daughter products showing the course of their biodegradation during incubation in these microcosms. And as you can see, each one is eventually biodegraded with the accumulation of ethene. Now, what about the biodegradation of these daughter products? This is a little trickier because remember, as the daughter products are formed, they're initially much lighter than the parent. Because remember, there's that preference for carbon-12. So initially, there's going to be a lot more carbon-12, or let's say it differently, a lot less carbon-13 in the daughter than there was in the original parent. Now, if that daughter product then subsequently starts to degrade itself, it will start to get heavier too. So how can we tell if a daughter product has been or is degrading? Well, we use that same rule of thumb and we compare it back to the parent source. If the daughter product becomes heavier, in other words, the del 13C value is less negative than the parent source, again by 2 per mil, we consider that to be substantial evidence that the daughter product has also been degraded. So now we're looking at the del 13C values for the daughter products as well as the original PCE. And as you can see here, initially all of these daughter products are going to have del 13C values that are more negative, therefore the daughter products are lighter than the original parent. But then as they start to degrade, you see the del 13C content start to increase. So you've got this formation process going on that's basically feeding in light material, and a degradation process going on that is preferentially taking away light material. So the rule of thumb is this, again, remember our threshold value here. When the del 13C value of the daughter product exceeds the del 13C value of the parent source by at least 2 per mil, then we have convincing evidence that those that those daughter products are themselves being biodegraded. So let's consider some hypothetical field data. Let's suppose we've taken a sample and we've done analysis for PCE, TCE, cis-DCE, vinyl chloride, and ethene. And we've done isotopic analysis, and we've gotten the del 13C values that you see here. The question is, are they being biodegraded? If you go to the EPA guide, you'll find a diagram similar to this one. This diagram shows you that for manufactured sources of these various products, like take PCE for example, it shows you the range of del 13C values that are found in these manufactured products. So if you do not know, and we'll return to this in a moment, what the actual del 13C value is of your actual source, then you can go to literature like this, look at this range, and take the largest value, and in this case for PCE, it's minus 24.06. And then the question becomes, or then the rule of thumb becomes then, that if I see a del 13C value greater than the 24.06 by 2 per mil, by greater I mean less negative, in any of my field data, that can be taken as good evidence that biodegradation of PCE is taking place. So if I go then to my table again, remembering that that value for PCE was minus 24.06. If I add 2 per mil to that, that's minus 22.06. So anything greater or less negative than 22.06 represents biodegradation. So we had a value of minus 20 for PCE. So is it biodegrading? Yes. How about the TCE at minus 14.1? Remember, we're comparing it back to the original parent compound. That is more, has a higher del 13C value than 22.06. In other words, less negative. So yes, the TCE is definitely biodegrading. The same can be said about DCE. What about the vinyl chloride? Well, it has a value of minus 37.9. So that is very much depleted compared to the 2206. So even though vinyl chloride may be being degraded while it is being formed, this does not provide us with absolute evidence that vinyl chloride degradation is taking place. And the same can be said of ethene. Now ethene, the maximum value for ethene, by the way, is always going to be the del 13C value of the source. Now here's some similar data. Now we're just looking at a flow path now. And here we have a source area, and contaminants are flowing in this direction. In this well, again, let's say we're looking at PCE, minus 27. Does that indicate biodegradation? No. Minus 26.3, does that indicate biodegradation? No. Minus 17.8, does that indicate biodegradation? Yes, it does. Down here, monitoring well 6, minus 8.2. again, we see evidence of biodegradation. One of the things that can happen to you in the source area, particularly if there is NAPL available, the continued dissolution of the contaminants from an NAPL source can greatly confuse your isotopic data in the source region. So it's not uncommon that you're going to have to look down gradient to see those evidences of biodegradation. So in terms of summarizing our applications for CSIA, what advantages do we have? Well, again, we have a technique here that can, under conditions, provide conclusive evidence that biodegradation is taking place. And it is applicable to electron acceptors, as we were just seeing, unlike stable isotope probing. But it will also be applicable to electron donors. So CSIA is more widely applicable in terms of contaminant type than is SIP. It's also relatively inexpensive. There are some quantitative applications also of CSIA that we don't have time to talk about today, but I'm told are going to be the subject of a future webinar in about the March to April timeframe in 2017, where you will learn about how you can use CSIA to estimate the extent of biodegradation of a parent compound, and you can also gather evidence that can help you distinguish between biotic and abiotic mechanisms or aerobic versus anaerobic mechanisms. Now, one of the limitations, though, of CSIA is that not all degradative processes result in significant isotopic fractionation. So in some cases, it may be difficult to detect biodegradation simply because the del13C value is not changing very much as biodegradation takes place. And in those cases, BTEX is an example, you might have to have more than 50% degradation of the contaminant pool before you can conclusively say that biodegradation is taking place. And as I was indicating earlier too, fractionation can be masked, and the most common problem is that if you are near a NAPL source, then the fractionation that may be occurring in the aquifer is going to be basically invisible because of all the unfractionated material that is continually dissolving in the groundwater. So let's kind of go through maybe a little decision tree kind of discussion here on deciding when do I use SIP and when do I use CSIA. Well, the most important question to ask here initially is the contaminant being used as an electron acceptor rather than an electron donor? If the answer to that question is yes, then CSIA is the definite choice. Stable isotope probing cannot be used when the contaminant is being used as an electron acceptor. Your only choice there is compound-specific isotope analysis. For example, the kinds of compounds that we've been discussing here and added some other compounds to the list here. All these different chlorinated compounds that undergo reductive dechlorination. What if we want to evaluate abiotic degradation mechanisms? For example, using things like zero-valent iron or naturally occurring material that may be giving us abiotic reduction of contaminants, then again, CSIA is your only choice. With abiotic degradation, stable isotope probing is much less sensitive. Well, let's go then to compounds that are being used as carbon and energy source. Most of these compounds, your choice is going to be stable isotope probing. For example, things like naphthalene, PIHs, etc., PAHs rather. When you've got a large compound like this, it's hard to see very much fractionation during biodegradation. And in that case, then, stable isotope probing becomes your only choice for establishing biodegradation of the target compound or contaminant. What about BTEX or things like MTBE, or we could add TBA, etc.? Probably your best choice is going to still be stable isotope probing. Stable isotope probing, as we've already said, will work well in the presence of NAPL. It works well in the source area. It works well with high contaminant concentrations. All of these types of conditions can provide confusing results with CSIA. If we have a long or dilute plume, then CSIA could probably be used, though you probably want to do two-dimensional CSIA, doing both carbon and hydrogen isotopes. And as we've said before, if you would like to try to estimate the fraction of contaminant remaining or the fraction of contaminant that has been biodegraded, CSIA can provide you with that information. So with that, I'll conclude my remarks, and if we have time for questions, I'll be happy to try to address any of those.
Thank you very much, Kerry. That was a great presentation. And I sent a message to all of you that are attending. We uploaded some handouts to the GoToWebinar control panel. You should be able to access them on your control panel under handouts. And it's just some information that's on the Microbial Insights website that talks a little bit more in depth about CSIA and stable isotope probing. So I think they're a nice complement to what Kerry presented today. You will need to save these to your computer if you want them so you can access them in the future. You can also get all this information on our website. Okay, so thank you again, Kerry, for doing this. Let's move to the questions. The first question that we have is about stable isotope probing, and it's talking about the phospholipid fatty acids. We know that there's some 13C that's present naturally in the environment. So the question is, can we assume that all 13C in the lipids are associated with the probe that we're adding?
The short answer is no. You can assume that anything over 1.1% carbon-13 is associated with the probe. So depending upon how deep you want to get into the analysis, if you, for example, Microbial Insights can provide you with a more detailed analysis that shows you specifically what particular fatty acids carbon-13 was incorporated in. Anything over 1.1% represents incorporation from the probe, so that helps you determine which particular fatty acids are incorporating carbon-13 from the C13 labeled contaminant. In some cases, that can give you clues as to what kinds of organisms are actually involved in the degradation process. In some cases, it might be you can distinguish between more aerobes are participating versus more anaerobes, or you might see both groups participating. In some cases, there are specific PLFA markers that are indicative of, say, for example, certain sulfate-reducing bacteria. If you happen to see those particular markers with a large incorporation of carbon-13, it can indicate a significant participation from sulfate reducers. Okay,
great. All right, in the first CSIA example, what if the source also had a TCE product? So you have PCE and TCE being released. Would you compare the 13C to the original parent or to the TCE parent?
Well, that's going to require some information about the original source. If the original source contained both PCE and TCE, then you would compare your delta 13C values to what you might believe the original source material to be. Again, if you go to the EPA document, that would provide you with that kind of information. But if you think that the TCE in the source area actually came from degradation of PCE, then everything would be compared back to that particular del 13C value. I'm not sure if I'm answering the question here, but one thing it does bring up one thing that I did not mention that I intended to. The EPA document provides you with ranges of values of manufactured products. If you would like to be more precise, and you can actually obtain samples from your site from the source area that you do not believe have been biodegraded, then that can provide you a more precise value of the del 13C value of your ultimate parent compound. In some cases, rather than using the most enriched value in the EPA range, sometimes you can be a little bit more sensitive in teasing out degradation that way. But you've got to be sure that you are sampling a true source without biodegradation having taken place.
Okay, great. Do you feel like CSIA can be used to determine and distinguish multiple sources or releases?
Well, there's certainly been work like that has been done. I've not been involved in that, but certainly that's the case. I mean, just look at the EPA data, for example, and the ranges of the del values of different manufactured products. I think there is enough of a range there that you could get lucky and distinguish between two products. But if the del 13C values are quite different, as I said, you might be able to make that distinction. If they're close together, that doesn't necessarily mean that they're exactly the same or they're different.
Okay. And then do you mind if we make the slides available for this presentation?
No problem. Would you like me to send them to you, Dora, and then you can send them out? Or
yeah, that would be great. Yeah, if you can send them to me, then we'll attach it to the email that goes to everyone.
Yes.
You know, talking about multiple sources too, we can also do some two-dimensional or in some cases three-dimensional analysis to be able to help differentiate sources a little more clearly as well.
That's right. I'm glad you brought that up, because if you're looking at both carbon and chlorine, then you've got more resolution there.
Correct. Okay, and then talking about 1,4-dioxane, and this is one of the handouts that I uploaded. One of the questions is, what do we recommend for 1,4-dioxane? Do we look at CSIA or stable isotope probing?
My only experience with 1,4-dioxane is in stable isotope probing, and there have been several successful applications of SIP with 1,4-dioxane. So I would have confidence in recommending SIP.
Yeah, I agree. And it can be done with CSIA as well. There's just less information in literature about some of the fractionation and what happens with this compound. Okay. The next question is, when we're using stable isotope probing, how much effect is the microbial accumulation on the bead that promotes the biodegradation, which may not necessarily be there in the plume? So I guess the question is, you know, if we're seeing increased organisms colonizing the bead, are we going to see increased amounts of degradation with the SIP?
Well, I think there's a possibility that if the concentration of the contaminant is quite low in the aquifer, it may be below a threshold value that results in significant rates of biodegradation. In other words, it may be low enough that it's not providing enough of an energy source for the organisms to actually grow on. And you could have the effect of concentrating that compound in the beads and therefore showing biodegradation when biodegradation may be occurring much more slowly in the aquifer. But if that's the case, that would be a situation in which, again, the contaminant concentrations would be quite low. There are remedies out there that take advantage of that same effect of adding powdered activated carbon, for example, to an aquifer to concentrate the contaminant to increase rates of biodegradation. So whether that's the case or not, the benefit to you is you find out if the organisms that are there in the aquifer are capable of degrading that compound under the actual conditions that exist in that aquifer. If the concentration of the contaminant in the aqueous phase in the aquifer is very, very low, then it might steer you to a treatment method that allows you to concentrate that contaminant.
That's a good idea. Okay, Carrie, also on CSIA, what do you consider to be the lowest concentrations of the compounds that can be used, especially for chlorinated compounds? Do you have any rules with them?
I don't. I would refer that kind of question to Microbial Insights and as far as what their detection levels are.
Okay, so what we would say on that is, in general, for carbon and chlorine isotopes, we can detect approximately 1 part per billion for each of so 1 ppb for each of the chlorinated compounds. For hydrogen, it's a little bit higher, especially when we're talking about TCE, and it's 20 to 50 ppb for those. Okay.
Can I add to that for
Sure.
that response? If you do have very low concentrations of chlorinators in the aquifer, and you're afraid that you might be bumping up against those detection limits, one thing you could do is to use a biotrap in the aquifer to actually absorb and concentrate the chlorinated hydrocarbons to retrieve those for analysis.
Yeah, that's a good idea. Okay, so it's known that some chlorinated solvents can degrade abiotically over long periods of time. So can this change in the 13C value content of the residual compound be mistaken as evidence for recent biodegradation?
I suppose that is possible. We're a little bit outside of my experience here in terms of abiotic mechanisms, and I'm not sure I can answer that question. Dora, perhaps you could discuss that, but I think that your speaker for the March to April webinar on the quantitative applications could give you a much more definitive answer on that.
Yeah, I think what we could do is use some of these approaches, like looking over like the length of the plume, to be able to help us kind of understand a little bit more about the degradation or time series data to help us understand if it's current biodegradation versus something from decades with abiotic. Okay, the next question is, what are your thoughts on the application of these tools for co-metabolic processes, such as 1,4-dioxane?
Well, as far as stable isotope probing is concerned, well, I'll say right up front, I have no experience with CSIA and 1,4-dioxane and co-metabolic. But as far as the stable isotope probing is concerned, you'll still be able to detect the ultimate degradation of those labeled compounds from 1,4-dioxane. It's just through co-metabolism. It's just not going to you're not going to be able to discern the mechanism, but you will be able to detect biodegradation, which is ultimately going to take place after that, the initial co-metabolic events.
Right. Okay. And we have one last question, but I'm not sure that I understand it, so maybe the person asking this question can elaborate. They ask, what's a better indicator of the carbon, hydrogen, or chlorine isotopes? I guess maybe they're wanting to know which you would recommend doing first, kind of a tiered approach.
Well, I think if we're talking about chlorinated hydrocarbons, as I assume we are here, definitely you want to start with carbon and see if the carbon answers your question. That's going to be the most economical approach and the most likely to be successful unless you're under some conditions that are difficult to interpret. And then I would start looking at the other isotopes, if that's the question.
I think, yeah, I'm assuming that's what we're getting. If not, if you want to email me, we'll be glad to clarify that answer for you. I think that's all the questions that we have today. Thank you very much, Carrie. This was a great webinar. I think it answers a lot of questions. And as Carrie mentioned, we're going to be doing these monthly webinars again next year. And hopefully in March or April timeframe, we'll have one that talks a little bit more about the quantitative aspects of CSIA and what we can do, and some deeper knowledge about getting into the kind of the more deeper two-dimensional and three-dimensional analysis and what we can do there. So if you'll tune in, we'll make sure to email everyone about that. But again, thank you for your time. We know you're busy. We appreciate you participating in our webinar. The link to the recorded webinar will be sent out a little later this week, along with these handouts and the slides for anyone that's interested in getting those. If you have questions, feel free to email us at info@microbe.com. We'll get with Carrie or we'll answer them as best we can. Thank you again, Carrie, for your time.
Thank you.
All right, thank you. Bye-bye.
Bye.